Okay, so welcome to this next video on the activation of T cells. Okay, so far what we've seen is that an antigen-presenting cell will dock a fragment of an antigen on its MHC class II molecule, standing for Major Histocompatibility Complex class II. Okay, and that will be recognized by a T cell receptor here in pink, and every T cell has a slightly different T cell receptor, and that T cell receptor will bind onto different antigens basically. So this antigen presenting cell will have to look far and wide to find a T cell which has a T cell receptor which can actually bind to the antigen fragment that is docked in this MHC complex. Okay, then when it does find that, the CD4 will also bind to the MHC, and together the T cell receptor and the CD4 binding to this MHC antigen complex will then deliver signal 1 for activation of the T cell. Okay, and we also saw how that alone is not enough to activate T cells, and indeed if you just give a T cell that signal, if you don't present the co-stimulatory molecules, then this T cell will enter a state known as anergy, which is where it doesn't die, but it will never ever be allowed to stimulate, will never ever be activated ever in its life. It will live out its life and never be activated, even if another T cell comes along and provides both the MHC class II with the antigen and the co-stimulatory molecules. It won't divide. You can never stimulate an allergic T cell. Okay, and that's basically a protection against autoimmunity, because if the antigen-presenting cell is only presenting MHC class II with the antigen, then that means that the antigen-presenting cell hasn't been activated. Because remember, I told you that if a pathogen invades, it will have many pathogen-associated molecular patterns, PAMPs, on its in its structure basically, and this macrophage has receptors for PAMPs, and basically when those receptors for the PAMPs become activated, then the macrophage starts expressing these co-stimulatory molecules. So if it hasn't expressed those co-stimulatory molecules, it suggests basically that it's presenting an antigen that didn't come from some pathogen. Instead, it might be presenting an autoantigen basically. So that's a protection against autoimmune attack. Okay, right. So, however, if the antigen-presenting cell does deliver the co-stimulatory molecules, which are CD40, this green one, and then the orange can either be B7.1 or B7.2, which have other names. B7.1 is also called CD80, and B7.2 is also called CD86. Then those can bind to their respective receptors on the T cell and deliver signal 2 to that T cell. Now, okay, so we will now want to look at what's going to happen within the T cell when it receives both signal 1 and signal 2. Well, basically, we're going to jump a step because it's not known what actually happens. But when signal 1 and signal 2 come in, so here's signal 1 and signal 2, what is known is that if you deliver both of them together, you start allowing calcium in from the extracellular fluid into the cytoplasm of the T cell. So this is the T cell membrane, if you like, T cell membrane. And this up here is the extracellular space, and this down here is the intracellular space. But we don't know how this calcium actually gets into the cell, basically. We don't know what protein is involved in that. The other important thing is that this calcium is not just delivered in one great big sort of puff, basically. Instead, what you get is calcium oscillations, i.e., if we were to plot the calcium concentration, let's say we pick this spot here to put in our detector, and it's going to measure calcium concentration. So it's going to measure calcium concentration at that spot. What we would see if we plotted calcium concentration versus time is we'd see oscillations in the calcium level, like so. So you don't just see calcium going up and staying up, you see oscillations, basically. Okay, right. So, let's see what this calcium actually does within the T cell. Well, basically what happens is when calcium goes up, it binds to calmodulin. So let's just have a brief revision of the structure of calmodulin. So calmodulin is a protein that in cartoons is often drawn like so. Okay, so it has these two lobes, an N lobe and a C lobe. Okay, and both of these lobes of calmodulin have two calcium binding sites, okay? So each lobe of calmodulin has two calcium binding sites. And the specific type of calcium binding site that you have here is something known as an EF hand dimer, which is why you have two of them, basically. So let me just explain to you what an EF hand is, and then I'll explain what an EF hand dimer is. Okay, so an EF hand is basically just a domain of a protein or of a polypeptide which can bind calcium, and it has a structure that kind of looks like this. So if this line represents the polypeptide, basically, it has a loop like this. So this is the EF hand. And if you look at the amino acids that make up this polypeptide that within the loop here, so if we split it up into little amino acids, so these little sort of intervals I'm going to use to represent amino acid residues, okay? So this is a polypeptide, so it's amino acid after amino acid after amino acid, and these are now the amino acid residues. Okay, well if we look at the amino acid residues in this loop, what we find is that most of them are acidic residues. So to give an example, aspartate is an acidic residue. So let me just draw you the structure of aspartates. Okay, so this is the amino acid, well this is the molecular structure of the amino acid aspartate. Okay, so here we have the amino terminus, well the amino group of the amino acid aspartate. Here's the carboxyl group. Here's the alpha carbon here with the hydrogen off the alpha carbon. And here is the R group of aspartate. So this bit here. Now, as you can see, it has basically an ethanoic acid group sticking off the side. That is what I mean by an acidic residue. Okay, so why is this important, basically? Well, if you have acidic residues, what do acids do? They donate protons to things. In the case of aspartate, what it can do is it can let this proton go off and donate it to some other group. And what will that do? It will leave this oxygen with a negative charge on it, basically. So, what's important is that if you have lots of acidic residues here, they might all donate their protons off, and that means that these residues are going to acquire a negative charge. So really, you're going to have a bunch of negatively charged residues here. Now, calcium is a divalent cation. It has a double positive charge, so it's going to like to come and sit in this loop and be coordinated by all of these negatively charged acidic residues which have donated their proton away, basically. Okay, so that's pretty much what an EF hand domain is. Now, if you want to talk about what an EF hand dimer is, then an EF hand dimer is basically just two EF hands next to each other. So you have a linker region in between, and then you have another EF hand sitting like this. And actually what you find is that EF hands very rarely occur as monomers. They often do occur as dimers. So you often have two EF hand domains sitting next to each other. And this means that a calcium ion can come and sit in here, and a calcium ion can also come and sit in here. So you have two calcium binding sites basically sitting next to each other. Okay, right. So that's what you have in these two lobes of calmodulin. You have an EF hand dimer in the N lobe of calmodulin and an EF hand dimer in the C lobe of calmodulin. So when calmodulin has no calcium bound to it, i.e. when calcium is very low in the cytoplasm, then it is known as apocalmodulin, and that's often denoted apo CAM for calmodulin, CAM like so. Okay. Now, when calcium goes up in the cytoplasm, what happens is that calcium is going to bind in these calcium binding sites on the two lobes of calmodulin. So you've got 4 calcium binding sites, so overall you're going to get 4 calcium molecules, calcium ions, binding to the calmodulin. And what happens is that when the calcium binds to the calmodulin, calmodulin changes its conformation. Okay, so firstly it sort of uncoils. You can see I've sort of drawn it with this curvature to it, but it sort of straightens out. So the lobes sort of move outwards away from one another. And this polypeptide that links them here, it was linear basically, but what happens is that when the calcium binds to the calcium binding sites, that linker takes on the structure of an alpha helix. You get this alpha helix, which is now linking the two lobes of calmodulin together. So here is calmodulin now linked by these by this alpha helix basically here. So this is an alpha helix, a secondary protein structure. Okay, and this is the structure that it has when there is calcium bound to these 4 calcium binding sites. Okay, so this structure, which is calmodulin with 4 calciums bound to it, this is known as a calcium calmodulin complex, basically. So, or just calcium calmodulin, or people will often just denote it Ca2 plus CAM for calcium calmodulin. Right, okay, so when calcium goes up on one of these peaks here, what's going to happen is that the calcium is going to bind to the 4 calcium binding sites of apocalmodulin and convert it into a calcium calmodulin complex. So you're going to get lots of calcium calmodulin complexes forming in the cytoplasm of your T cell. Okay, now, what do calcium calmodulin complexes do? Well, basically, they can bind to an enzyme and activate it. Okay? And what enzyme am I talking about? Well, I'm talking about an enzyme known as calcineurin. Okay, so this enzyme is calcineurin. Okay, and often it's denoted CN. So if you ever see CN, that often will denote calcineurin. And basically, when you get these calcium calmodulin complexes forming, these calcium calmodulin complexes are going to bind to calcineurin, so here it is, binding to calcineurin, and they are going to activate the calcineurin, basically. Okay, and we'll continue this discussion in the next video.